Engaging. From a distance, we see skin and bone. But zoom in far enough, and the human body reveals itself as a biological data center, a system built on dense streams of information. Every day, we generate petabytes of raw data. Our DNA sequences, the specific proteins within our cells, and chemical signatures of our microbiome all produce a constant, massive output of biological code. This volume of information creates a mathematical wall. The human brain cannot manually scan billions of genetic base pairs to find the single mutation responsible for a rare disease. Organizing and interpreting this biological noise requires a specific computational framework, bioinformatics. In 2026, understanding human biology is a high-stakes logistics operation. The primary barrier to medical progress is no longer a lack of information, but the speed at which we can process it. Bioinformatics functions at the precise intersection of three fields: biology, computer science, and mathematics. By integrating these disciplines, researchers can manage the heavy lifting of biological data at scale. This work follows a strict 5-step pipeline. It begins with data generation, capturing a patient's biological samples, and moves to data processing, where raw, fragmented code is cleaned and organized into a standardized format. During the third phase, analysis and modeling, statistical tools scan those organized rows to detect hidden patterns or anomalies that indicate a specific health risk. Finally, through interpretation and applications, these patterns are translated into clinical decisions. This is where a data insight becomes a physical treatment, such as a drug designed to target a specific protein. This pipeline provides a reliable, repeatable method for converting abstract code into the chemical compounds used in modern medicine. By 2026, artificial intelligence has become the primary accelerant for this entire data pipeline. Using foundation models and large language models, researchers can instantly cross-reference a new genomic sequence against the entirety of existing scientific literature, identifying relevant studies in seconds. We are also deploying generative AI models to design novel drug molecules. Instead of relying on years of physical lab trials, the system calculates the exact molecular geometry needed to bind to a target, bypassing traditional trial and error. This technology doesn't replace the researcher, it scales human discovery, allowing scientists to work with a speed and accuracy that was previously impossible. We can see the impact of these tools in a clinical setting by looking at how a patient in 2026 receives a customized treatment plan. The process begins with whole genome sequencing, where the patient's entire DNA structure is digitally mapped. Next, a process called variant calling identifies the exact genetic flaw. The system scans billions of base pairs to isolate the specific mismatch. The AI then interprets this variant to recommend a tailored therapy. This direct link between data and treatment is precision medicine. To execute this workflow, we rely on a specialized toolkit: programming in Python and R, navigating databases like NCBI, and structural tools like AlphaFold 3. By integrating code with biology, we are actively moving toward a new healthcare model, one defined by data-driven prediction. In 2026, the distance between identifying a raw genetic sequence and delivering a life-saving therapy is shorter than it has ever been.